Hello and welcome to Akoya Biosciences and Proscia's joint webinar, From Research to Translational Medicine, Changing the Language of High-Parameter Quantitative Tissue Analysis. My name is Nathan Bookbinder. I'm the Chief Product Officer at Proscia, a digital pathology and artificial intelligence software company, and I'm joined today by my partner and colleague Dan Eversole, the Product Manager for FinOptix software at Akoya Biosciences, the multiplex immunofluorescence and biomarker discovery company. We're excited to be here today to talk about the changing landscape in immuno-oncology and immunofluorescence, the new technology that's advanced in the industry, and the novel ways that a best-in-class image analysis solution can be paired with a best-in-class management, collaboration, and workflow solution to drive meaningful changes in how research is conducted from benchtop to bedside. Just to note, neither Akoya nor Proscia's products have been reviewed or approved by the FDA. And a second note, we'll be holding questions until the end. If you have a question, kindly type it in the chat and we'll be sure to get to it in the time that we've reserved for Q&A. So let's dive right in. To most of you on this webinar, it should come as no surprise that immuno-oncology is big. It's big both in terms of the capacity that it has to shift the revenue and sales of new and exciting products that make their way into the market, but it's also big in terms of how it's changing the standard with regards to the kind of research that's being done at large academic centers, as well as in pharma companies, CROs, and other biopharma research. And really, at its core, this immuno-oncology wave is focused on the concept of targeted therapies and targeted selection of patients. It all ties back to personalized medicine, the notion that each individual responds slightly differently to any candidate, and that throughout the entire R&D process, from patient identification and patient stratification to clinical trial recruitment to the development of companion diagnostics and the end products themselves, the ability to better understand and assess and interpret and quantify the likelihood of response to a therapy is massively important in dictating what products make their way to market and how they're marketed. And fundamentally, all of this starts with the challenges prevalent throughout immunohistochemistry. Now, immunohistochemistry can be a very powerful tool, but it's wrought with problems, and the central problem is that it is subjective. When you look at this image, you might have 4 different colors, 4 different resolutions or bins that you're able to classify any given cell, structure, or other object into. The challenge is that actually describing this image is qualitative. It's very hard to define distinct patterns, and therefore the reproducibility is poor. That leads to statistically questionable data and results as I conduct a large-scale study, and it also makes the process cumbersome and time-consuming. So take, for example, the response to Keytruda in previously tested non-small cell lung cancer patients. Even amongst the group of PD-L1 positive individuals, only 41% were responders, while 59% were non-responders. And in PD-L1 negative individuals, 13% were responders, while 87% were non-responders. So what does this mean? It means that more than half of patients testing positive for the PD-L1 biomarker didn't respond to the treatment. It's an imperfect immunohistochemical method of determining likelihood of response. And that results in overall poor predictive values for demonstrating the benefits of many of these novel therapies. And so increasingly, multiplexing has become the solution to address that problems. It is quantifiable. It comes in the form of distinct patterns that are easy to detect, easy to measure, very reproducible, and it allows all of the analysis from a single slide, which means that you can actually ensure that the variability is minimized as you're conducting this high parameter research. Now, while immunofluorescence and multispectral imaging have taken an enormous step forward over the past couple of decades, what we're finding is that there's an increasing challenge associated with managing all of that data that's coming out of that pipeline. A single image could be a gigabyte or more large. These images are challenging to manage. You're creating hundreds, thousands, or more of these in a single study. And often these studies are collaborative in nature. They're global. You have to be able to transmit this information between multiple sites, either in the same country or between countries. And that poses a real technical and infrastructural challenge to many organizations that previously had pathology center around physical glass or at most whole slide imaging of bright field images. And so what we're seeing is that as organizations, as research organizations shift towards higher throughput analysis pipelines that are integrated and a more core part of the research that they're performing, there's a real need to blend and merge the outputs of high throughput, high parameter analytics with critical data management and image management systems. And so to start to talk about what Akoya and Proscia are doing together to address these challenges, I'd like to hand it over to my colleague Dan Eversole, who'll go into a little bit more detail around the work that Akoya does.
Thank you, Nathan, and thank you to all attending today's webinar. I'd like to take the next few minutes to introduce you to Quoia Biosciences before walking through our FinOptix platform, specifically our spatial biomarker analysis tools. Quoia Biosciences is the multiplex immunofluorescence and biomarker discovery company. Our goal is to provide the capability, functionality, and support for the identification of biomarkers that will enable precision immunotherapy through spatial biomarker discovery. This will be enabled across the continuum, from early discovery through clinical deployment. While our focus is the field of immuno-oncology, spatial biomarker discovery is extremely important in a whole host of other fields, including neuro, transplant, and cardiovascular. In the early biomarker discovery phase, learning about underlying biology within intact tissue sections is key. Researchers need high plexing capabilities, such that they can visualize and quantitatively assess many different cell types and protein expression levels. Once the biology related to disease progression or a drug's mechanism of action is understood, high-plex assays are boiled down in the clinical trial phase to something that is high throughput, standardized, and reproducible. If a biomarker proves to be valuable during clinical trials in either predicting response or revealing a drug's mechanism of action, it is then commercialized into a clinical platform, such as a companion diagnostic. Akoya offers two platforms, the Codex and FinOptix platforms that drive your research across the continuum. Our Codex platform specifically is designed to meet your needs in early biomarker discovery. It provides the capability for ultra-high parameter biomarker discovery, enabling up to 50 markers per slide. It is designed to fit your budget by integrating with existing microscopes such as the Quantitative and Leica. Furthermore, it enables simplified workflows via load-and-go automated data acquisition protocols. FinOptix, on the other hand, while it can be used in the discovery phase, it is designed to take a researcher through the translational and clinical pathway. It is designed to provide high throughput data collection, and results are battle tested, proving in numerous peer-reviewed studies to be accurate and reproducible. With that, I'd like to continue the discussion only on the FinOptix portfolio. FinOptix is an end-to-end platform providing solutions for multiplexed immunostaining using the Opal fluorophores, multispectral imaging with Mantra and Vectra quantitative pathology imaging systems, and multispectral unmixing analysis with Inform and our new Finopter and Finopter Report R scripts. These solutions enable a workflow that permits the simultaneous visualization and quantification of multiple cellular phenotypes in an intact tissue section, enabling deeper understanding of cell-to-cell biology and overall phenotypic architecture in the tumor microenvironment. This has the potential to enable researchers to develop new diagnostic tools to predict immunological responses in cancer patients. Multiplex immunofluorescence enables researchers to assess multiple phenotypes. Multispectral imaging enables the ability to unmix fluorescent signals from both one another, which reduces spectral crosstalk between the fluors, and from the background autofluorescence innate to the tissue. This means you can have the confidence the signal you want to measure is truly from the signal of interest, regardless if signals are co-localized. These are all important factors to confidently assessing tissue quantitatively, not qualitatively. Here in this image, we show a 6-plex, 7-color multispectral image of breast cancer. You can see how spectral unmixing leads to well-isolated signals from both immune cells and tumor. And this can be found here along the perimeter of the image. You can see the single marker views, which added together represent the area outlined by the white box. For context, the 6-plex assay can enable the analysis of up to 63 different phenotypes in a single tissue section, which is important when attempting to glean as much data as possible from precious clinical samples. Here in this slide, we show how the autofluorescence signal can be isolated and unmixed from the signal of interest. In the PD-L1 negative sample, you can clearly see the autofluorescence signal in the spectrally mixed image. That can be found here on the left-hand side, middle row. When unmixed, the background goes virtually zero. So if we take a look down at the bottom left-hand corner, you can see that after unmixing, the signal of the autofluorescence went to zero. This means with it at zero and completely removed from the signal of interest, you can with confidence analyze the PD-L1 expression in both low and high expressing tissues. So what makes this possible? Akoya has a suite of tissue analysis softwares, including the following: PhenoChart, which allows you to look at the tissue as a whole slide context, and it allows you to do annotations for downstream analysis. Inform Tissue Finder provides whole slide spectral unmixing with tissue analysis and phenotyping. FinOptix is our authourable spatial analysis scripts, and our brand new FinOptix reports provides very nice ways to look at complex phenotypic analysis, allows you to do spatial metrics between the different types of phenotypes, and you can look at staining quality across the different components within your image. All right, now that we know what software goes into making this happen, let's look at the workflow that's associated with this. So the question comes, how does this all come together to enable the quantitative assessment of phenotypes across the entire tissue section? So to do this, let's specifically look at our new motif whole slide multispectral workflow. So a customer will stain their slide with Opal Polaris kits, and then they will go ahead and scan this slide with the Polaris. The image that comes off the Polaris can be opened in PhenoChart, and it's already unmixed. So in PhenoChart, you can look at all the individual discrete channels, including the autofluorescence, which has been isolated. It also enables you to look at the different pathology views, which are our simulated IHC views, or bright field views of the fluorescent stains. From there, you can do your annotations and choose which regions you want to import into Inform for analysis. Inform analysis will do the spectral unmixing and tissue, cell sag, phenotyping, and thresholding, which can then be output to a variety of programs of your choice. 3 that we are offering is FinOptix for your spatial analysis, FinOptix reports to do your data analytics, and soon we're going to be adding tools for whole slide exploration and analysis. So to start, I want to take our attention to Inform and walk you through what an Inform analysis would look like and what are the outputs that come from it. So here you can see an image of a colon cancer. This would be a standard RGB image that you would get from any sort of microscope, regardless if it's our scope, Leica, Keonce. And what you'll notice in this view is that the fluorophore signals are pretty much indistinguishable from one another. This is due to lots of bleed through between the different filter cubes in collection of the image. So this unmixed image can then run through our Inform this mixed image, can then run through our Inform unmixing engine. to begin to isolate those individual signals. So here you can see what an unmixed multispectral image with autofluorescence looks like. What you'll notice is that all the individual cells are becoming more visible, but there seems to be a very nice white haze across the entire image. This is very much due to the autofluorescence that's pervasive through this image, and it has a tendency to mask very important signal that you want to be able to analyze. So if I remove everything except the autofluorescence, here you get a really nice view of what that background autofluorescence signal looks like. So you can see it's very much pervasive across the entire image. And in the cases where you get the very bright white dots, that could be red blood cells, and many times the signal can overlap your signals of interest, and so you want to be able to isolate that and remove it. And when you do that, this is what the image looks like. So you can see it's got very nice contrast, and you can very much easily see all the individual cells. Furthermore, all these signals are very well balanced due to the use of tyramide signal amplification. And so what this means is you can take a very low expressing marker and you can balance it with a very high expressing marker. So in this image, some of our immune cells might have very low expressions, and so you want to make sure that you can be able to pick up those phenotypes with confidence, regardless if there's only tens of them versus the hundreds of cancer cells that are within this image here that very highly express the pancytokeratin marker. And so by balancing that, you have the confidence that you can do the phenotyping downstream. So the first step after unmixing, many researchers like to segment their tissues. And in segmentation, this is identifying the different regions and morphologies. So here I show a very simple segmentation of stroma and tumor, where the stroma is in green and the tumor is in red. After you've done your tissue segmentation, many times folks like to go ahead and cell seg. And so this is providing unique cell IDs for all the individual cells in your tissue. It allows you to then begin to identify the individual compartments within a cell, such as the nucleus, the cytoplasm, and the membrane. And so you can see that here in the image, where all the nuclear signals are green within the sample. You have the membrane stain in red, and then you have a variety of colors surrounding the cells. That is the cytoplasmic signal that's been identified. And so we have a technique called adaptive cell segmentation that allows you to accommodate for both heterogeneity within the nuclear staining, but then also accommodate for packing of cells due to inflammation or just clustering. And so you can begin to use all of your stains to assist your nuclear segmentation. This way you can be sure that you've identified all the cells within your tissue section. In addition to identifying all the cells within the tissue section, you can associate with more accuracy where those stains are located within the individual cell compartments. So once you have identified each of these cells uniquely, then you can begin to say what their phenotypes are. So you now have unique cell IDs for each of the cells. They have a location map on the tissue section, and you've begun to identify where the individual fluorescent markers are within the compartments of the cells, and you can use that to determine what the different phenotypes are. These can be single phenotypes, these can be advanced phenotypes of 2, 3, or more markers per cell. And so here I show a very simple phenotyping scheme where we have in green CD68 positive and PDL1 positive macrophages. So these are exhausted macrophages. In blue, we have CD8 positive cytotoxic T cells. In cyan, we have our pancytokeratin positive cancer cells. And in red, basically every other cell that's on the tissue. So we can bin everything else there. And really the concept is it's just a very simple machine learning technique that allows you to identify individual and complex phenotypes. So with very minimal training, you can come up with a very nice plot of all the phenotypes within the cells. Furthermore, because you've used multispectral unmixing, because you have balanced all your signals, you can with confidence identify phenotypes arising from low expressing epitopes, and you can count low phenotypic expressors. So regardless of how much stain there is on the tissue, regardless of how many cells there are of a phenotype, you can with confidence know that you are quantitatively measuring and counting those cells within your analysis. And finally, the last thing that can be output from Inform is our cellular positivity and cellular scoring. And so here I show a very simple positivity thresholding of PD-L1 in the stroma. And so you can see here in the blue and the red and the yellow where it's identified positive PD-L1 signal. If you remember back to the last image, this corresponds to where those exhausted macrophages were. They were the green cells that we were seeing previously. And so reason folks might like to do this positivity thresholding is because expression markers, specific your PD-L1 or your PD-1 stain, many times they have varying intensities across other cells. You might have completeness of stain issues, and so it doesn't lend itself as nicely to doing phenotyping, and so you might have a very large range of expressions of those markers, and so thresholding and cellular scoring provides a better picture of what that can look like. And so you can begin to bin those individual thresholds or individual positivity into bins of 0, 1, 2, 3, depending on how intense the signal is. And so this can be converted into an H-score, and the higher that H-score is, the more positive that sample is for that expression marker. So with that is all the Inform outputs, and we can move on to what do you do with all that data? How do you utilize that to truly understand what is happening in the tissue on the macro tissue architectural scale? So the first step, with each cell phenotypically identified, and now that we've given location coordinates along the tissue, it is now possible to study spatial relationships between individual cellular phenotypes. So here with FinOptix, we have created authorizable spatial analysis scripts to perform nearest neighbor phenotypic analysis, count within a radius, said another way, proximity measurements of one cell type to another cell type, and touching cells, so cells that share a border. These analyses can elucidate phenotypic interplays in addition to understanding whether a cell is in a functionally active state. So here you can see on the right-hand side 2 examples of what this can look like in FinOptix. And these are 2 default views within our FinOptix scripts of today. And you can see the nearest neighbor analysis in the upper right-hand corner where you have the ball and stick model between 2 different phenotypes. And here in the lower left-hand corner, you can see the touching cells, and cells that aren't touching aren't filled in, and the cells that are touching have filled in, and the 2 different colors is the 2 different phenotypes associated with those cells. Now, why is this important? So it's all about spatial metrics. You really want to be able to understand how this interplay is affecting your drug's mechanism of action, or you want to be able to understand what is the interplay within the biology. So here's one example that we've done an analysis with FinOptix. This was done, if I remember correctly, in a melanoma section. Here the annotation was hand-drawn by a pathologist along the normal tumor interface, and what they did is they looked at the different cell expressions across that interface, so CD31, CD8, PD-L1, and Tregs. And so what you can see in the PD-L1 positive graph, we have an immune exclusion. So what this means is the tumor cells are expressing PD-L1. This is creating a blockade. The infiltrates are stuck outside the tumor and are unable to infiltrate into the tumor doing their job. But once you've knocked down that PD-L1, which you can see here in the bottom graph, the immune infiltration can occur. And now all of those immune markers are within the tissue. So this is just one example of what you can get from spatial metrics, but this is a very important example within the immuno-oncology world of today. All right, so what's next? What's next with FinOptix? So we are working really hard to bring you new analytical functionality to our analysis platform. So I really want to discuss next different examples of how our platform will enable assessment over the entire tumor microenvironment. We understand that fields of view aren't going to cut it moving forward. You really need to be able to understand what it looks like across the entire slide or TMA sections. So how are we envisioning doing this? Here's a few examples. So first I want to look at our heat maps of phenotypic densities. This example shows what an immune exclusion could look like across the tissue section from a high-level view. The spectrally unmixed 7-color motif image of PD-L1 positive lung cancer is shown on the left. To the right is a heat map of tumor marker density with the higher densities portrayed in red and white. If I take the next view, what we can see along the edges of the tumor are high levels of PD-L1 expression shown here in this heat map. So you can see along the rim of the cancer, you're getting these edges of PD-L1 expression. Moving further out from the tumor edge, you can see that anti-tumor T cells are accumulating at the tumor site, but are failing to efficiently infiltrate. So here we have an example of FOXP3, and we have an example of the CD8. So heat maps provide a really nice way to show a high-level visualization of the tumor microenvironment architecture. Next, we can assess phenotypic densities across the tumor stroma interface. So if we look in the region within the white box here, we can provide measurements of the localization of each phenotype. And so what we have here in white is the interface between the pancytokeratin positive cells and the stroma outside of that. And we can, across that border, look at the densities that are there. And so what you can see that there's very much a lot of agreement with those heat maps that I was showing previously. Within the tumor, you have high expression of the pancytokeratin. Along the edge, you have the PD-L1 expression, and then you have your FOXP3 and CD8 anti-tumor T cells located out in the stroma, enabled to infiltrate the tumor cells. All right, so while such density plots show an aggravated view of phenotypic densities across the border, they don't show the complete picture. So here we provide another example of an unmixed 7-color motif image of melanoma. The annotated line was hand-drawn along the normal tumor margin as identified by a pathologist. And if we separate analysis into equal area buckets along this annotation, so that's what this image here is showing. We've created equal area measurements along this annotated border. We can begin to really understand what the phenotypic heterogeneity looks like along the entire length of the annotated region. And so that's what's being showed in these plots here. So this arrow indicates the length of the annotated line here, and what we can see is the expression of different phenotypes along this annotated margin. So we have CD68, you can see the CD8, the CD8 PD-1 positive cells, and so forth. Where again, the red is really showing very high expressing markers. So this gives you a little bit more detail than what you would get if you just looked at the aggregated densities across that interface, which is shown here in the graph on the right. So this provides the researcher with a few different ways of looking at phenotypic heterogeneity within the tumor microenvironment. So you can begin to really get a nice, better completeness of picture of your tumor architecture, your tumor microenvironment architecture. To wrap up the FinOptix portion of the webinar, I leave you with a few parting words. FinOptix is about enabling quantitative analysis across the whole slide. While multiplex immunofluorescence permits staining of multiple markers in the tissue section, multispectral unmixing enables you to confidently visualize and identify all the stained cellular phenotypes and then reliably and accurately assess their spatial and functional states. Heat maps provide guidance for identifying important regions of interest within a tissue section and enable easy tools for understanding global phenotypic architecture and phenotypic interactions across annotated margins. With that, I'd like to hand it back to my colleague Nathan, who will next demonstrate the Concentric platform. This tool is about leveraging data beyond the glass slide to achieve faster, automated workflows and better outcomes. Like Akoya, they are turning pixels into patterns and insights that will change how we address the immuno-oncology market going forward.
Thanks, Dan. So while Dan went over what Akoya does with regards to its technology in the high-throughput, high-parameter analysis space, I'd like to spend a brief moment talking a little bit about what Proscia does in the digital pathology realm with regards to data and workflow management, as well as how we start to tie in the results and the outputs of the Akoya platforms into a digital pathology system that scales for the organization. Our digital pathology platform is called Concentric. There are 3 editions, but with regards to high throughput, high parameter analysis in the life sciences space, we have an edition of Concentric that's specifically geared to these organizations, that fuels projects and programs and studies that are templatable, reproducible, that need to be shared, administrated by a wide range of individuals with user controls and user permissions. So the Concentric platform actually serves as an operating system that the laboratory uses to manage not just the high parameter analysis results and multispectral imaging, but any kind of imaging that's going through the laboratory. There are a couple of core functional areas of the Concentric Life Sciences edition. The first is enabling collaboration and sharing. So Concentric can be configured to enable enterprises with multiple sites, literally any number of sites, to centralize their pathology data and access it either simultaneously through a real-time virtual multi-head microscope-like experience that allows any number of users to access these images all at once, or through collaborative sharing. Think of shared folders, shared programs and projects with the appropriate permissions and controls that enable this to serve the capacity that it needs to as a central platform for image management. We also host our own and third-party image analysis algorithms, and Akoya's software suite is one great example of how an open API allows Concentric to serve as the central platform, the operating system for an image-based workflow. We also have core critical infrastructure components related to data and image management that allow for the mass import or export of data from any source, automating as much of that process as possible, archiving of inactive programs or studies, essentially finding every way possible to make this kind of large-scale big data research as feasible and as cost-effective as possible. We have integrations into this edition for clinical trial management systems, for laboratory information management systems, for electronic laboratory notebooks, and for any other kind of third-party application, including analysis, that ties all of those data sources into one platform and allows for bi-directional transmission of information so that your pathology research is remaining connected to the broader clinical trial or research that you're conducting. In addition, Concentric for the life sciences is available on cloud or on premises, accommodating any user's requirements from the IT side. There's, of course, the basic viewer with robust sets of functionality for search, global search based off of any facet or any number of parameters, and the ability to annotate images in any number of ways, which again ties into this concept of creating ground truth data for high throughput and high parameter analysis. And then, like I mentioned before, all of this is hosted on a platform that enables a secure, intuitive experience with roles and permissions and audit logs that enable you to ensure that you're meeting the standards that your organization has in terms of how research gets conducted. So there are a couple of key ways that an organization might use Concentric for the life sciences. including multi-site program or study management, studies of any size involving any number of data sources, archiving and automated data lifecycle management so that you're appropriately and automatically accommodating the different research lifespans that exist for any of the programs and studies that you're conducting, and doing so in a way that's cost effective to your organization. And then, a whole range of functionality around how image analysis, including multispectral imaging and multispectral image analysis, gets integrated into the platform in a way that allows it to be shared with any other system that's a part of your study, and in a way that allows it to be shared with any other user, pathologist, researcher, oncologist, or other individual who needs to access that data. And for just a very quick minute, I want to really highlight how something like this might be applied, in this instance at one of our customers, Neuroscience Associates, a mid-sized CRO focused on neuro research and neurology-related studies. So for them, they had already adopted some level of digital pathology, some level of whole slide imaging. They're a preclinical CRO, and they were shipping hard drives full of image data. And you can only imagine that they were doing this with bright field imaging. If somebody was doing this with an enormous amount of multispectral images and analysis results, it would be untenable to continue going the hardware route. And so what Proscia did, what Concentric does for this lab, is provide a management platform that enables them to share large digital pathology data sets with their own clients, with the pharma companies that they served, with other academic sites for whom they were conducting research. And that concept of centralizing all of the images, automating the process of making it accessible to the end users, and doing so in a way that scales globally, regardless of how many sites were involved in these studies, made a world of difference to NSA, and we're excited about how we can grow the platform with groups like them. But if we think about this again from the perspective of what Akoya is providing, Akoya's cutting-edge technology really outputs three core elements that tie into the Concentric platform, and those are the spatial analyses, the data reporting, and the image exploration and analysis tools. And so what I'm going to show you is what this might look like in Concentric. So when I look at this, what I'm actually seeing in Concentric is this multispectral image. This happens to be a melanoma image that has been stained with, I believe, 6 or 7 different immunofluorescent markers. And what I'm able to do is not just view the image and not just annotate the image and perform a wide range of various functions that you can see on the top toolbar, but I can actually start to organize all of these images as a part of studies, as a part of programs and projects that I can keep secure, keep as part of my repositories and my programs, or share them with any number of individuals. So in this instance, I've given one of my colleagues, Elby, edit permission, and a whole range of my other reviewers view-only permission. And you can use something like this to create massive data sets of multispectral images coming straight from the Akoya platform, particularly as the Akoya team incorporates more and more automation into the core platforms that they're delivering. We create an end-to-end seamless pipeline of high parameter analysis that ties into not just the fundamental elements of how research is conducted, but also how that research gets shared with any number of collaborators from around the world and with any number of study participants. So this kind of integration is actually available today. We're expanding on it, and we're excited to see how organizations are putting this to use to conduct really cutting-edge and breakthrough research. We're now happy to answer any questions that you might have about what Proscia and Akoya are doing together to change the landscape of high-parameter image analysis and multispectral imaging.